Since 1968, the New England Aquarium has provided care for thousands of stranded marine animals, most notably the endangered and threatened sea turtles. For some cold, stunned, or injured sea turtles, a stay at the New England Aquarium is in order before they return to their home in the sea. Through medical attention and rehabilitation, many sea turtles have been nursed back to health and released back into the wild. This evening, we have two very important staff members that lead their teams in this valiant effort. Connie, Director of Marine Animal Research, Rescue and Rehabilitation, has been at the aquarium for over 15 years. Her expertise with both marine animals and sea turtles has taken her around the world, and most notably, recently to the Gulf of Mexico to help with the deep water oil spill last year. Dr. Charles Innis is Director of Animal Health and has been at the aquarium since 2005. In addition to his work here, he also holds an adjunct teaching position at Tufts University, the University of Connecticut, and Mount Ida College. He is past president of the Association of Reptilian and Amphibian Veterinarians, as well as a member of a number of other professional organizations worldwide. Please help me welcome our lecturers for this evening.
Go ahead and get started, and we'll jump right in. I'm going to talk mostly about sort of general husbandry of sea turtles. We're going to go over some of the common species that we see around here and what we do to take care of them. And then Dr. Innis is going to come up and talk to you in detail about some of the clinical techniques and rehab methods that we use. So just to kind of give you a quick idea of the stranding program, we cover quite a bit of territory. It's over 1,000 miles worth of territory that we have to cover. And we certainly would not be able to do any of that without our volunteers. So it's basically responding to injured and stranded animals, also responding to a hotline. Sometimes that just means educating people that it's a resting seal. That's not the case with turtles. In our area, they don't rest on shore. But so it's a lot of fielding calls, educating the public, and responding in the field. And we do the majority of that with 5 full-time staff, 2 part-time staff that we bring on for turtle season. You'll see why in a few minutes. And of course, we use a lot of resources from our animal health department, as you'll hear from Charlie in the next few slides. So we've, in the old days, years ago, we used to do a lot of rehab of harbor seals, and we even rehabbed some dolphin species, white-sided dolphins. I think before my time, they may have tried some common dolphins, and they did a few pilot whales. But none of those species are endangered. So what we're focusing on now is endangered and threatened species, and those are the sea turtles. So the aquarium has recently invested in what we call the aquarium's Animal Care Center, and that's located in Quincy, Mass, and that's where the rescue department works out of. So we wanted to give you just kind of a sneak peek behind the scenes tour here. This is one of our turtle tanks. It's 24 feet long by, I think, about 10 feet wide. And you'll see some subdividing little devices that we put in there, and it depends on how many turtles we get and if there's aggression between turtles. So this is one of our, right now that's our largest tank. Back here you can see an empty space is a little tank right here, but in a year or two, once we're done with the giant ocean tank project, we're going to insert a tank that's even larger than the one you see here. So we are still building in Quincy, and we are still growing there. On the other side of the turtle hospital is another tank. This is a 15-foot, and we cleverly named it 15. It's easier for our volunteers if we name it by the length. And then we have some small isolation systems over here. These are all self-contained. There's no shared water, and that way if there's an animal that might have an injury, perhaps maybe it has a fracture, we don't want it in the general population, swimming into other animals or competing for food, then that's a great place. We also use these, we'll talk about these again in a little bit. We use those when the turtles first come in. They're not strong enough to swim, so oftentimes we have to give them supervised swims, and we take turns in these smaller pools so we can get a little bit of depth, but they can't really go too far and use up a lot of their energy. Oh, I forgot to give a shout out to our facilities department. Certainly some of the important things we're going to be talking about when we talk about husbandry and animal care is just keeping the environment clean, and that includes the water in our area. All our creatures live in it, so we have to test it daily and move it around. It gets brought in from the harbor, and it goes into settling tanks and heating tanks and that sort of thing. So we're lucky enough to have a group of folks that helps us with all of that. So this is a slide to show you some of the common species that we work with. This, this here is a green sea turtle. If any of you have been in the main building and seen myrtle, this is a tiny mini version of myrtle. This is what she looked like one day. I know it's hard to believe. But the green turtles come up here in the summer like the others, and they're a little bit different as they get older. At this life stage, they're probably about 2 in the size you see here. As they get older, they tend to go from a diet of primarily meat, so fish and squid, that sort of thing, to exclusively vegetation. They become herbivores. So it's actually really cool. The next turtle here in the upper right-hand corner, that's a Kemp's Ridley, and that's the most common turtle that we get stranded in our region. And that's also the most endangered turtle in the world right now. Again, this same sort of size class, it's a juvenile. It's probably about 2 years old. Currently, right now in science, we don't have an accurate method to age sea turtles. So it's an estimate based on what research has been done. Down here we have a loggerhead. This is one of our patients right now. And these turtles are quite large, and they have a very, you see how his beak overlaps here. Some of their favorite foods are crustaceans, so they have we have to be very careful when we're working with them because they could probably take your finger off. They like horseshoe crabs and things like that they crush when they eat. And we're going to talk a little bit more about their diets in just a few minutes. And then this animal down here, we're going to be talking about cold stunning, but not when we refer to the leatherback. So this is the largest of the marine turtles, and they are in our waters. But again, they don't cold stun like the others. So you're going to hear Dr. Innis talking about some procedures that we do on the cold stun sea turtles, and those are the turtles that get hypothermic and end up in our hospital. But again, the leatherback situation is a little different. We're going to get into that for you shortly. Okay, so it's important whenever we're talking about sea turtles, we like to talk about mass Audubon. All of the turtles that come here, I should say almost all of the turtles that come here, with the exception of a few, come from the Cape Cod area. So right in here, primarily, they almost always come from the inside of the Cape. Any of the animals on the outside have quick access, easy access back to the Gulf Stream, or to go south as the water temperature starts to get colder. That's sort of a trigger for them. They should go south, or they should go out to the Gulf Stream, wherever there's warmer water. It's not in here. So the Wellfleet Audubon staff actually holds the letter to go around and collect the stranded animals. And they have a fantastic program. They've been doing this, we've been working with them for 30 years, over 30 years. And they track the wind and the waves and the tides. And they have volunteers, everybody has assigned beaches, and the volunteers will go out and look for the turtles just after the high tide. And most folks ask us, why wouldn't you go at low tide? Because it'd be easier to find them. But as the tide recedes, it deposits the turtle on the beach, which then leaves it exposed to the elements. And we're talking the fall. Some of those nights, the temperatures drop down pretty cold, and that puts the animal in even a more grave situation than it was, if we picked it up 6 hours ago, or whenever the last tide was. So they actually walk the beaches at every high tide, even if the high tide's at 2 o'clock in the morning, 3 o'clock in the morning. It's pretty labor intensive for them, but it certainly makes our job easier. So I put a little picture in here just so you could see. This was 1999. We had 144 turtles, I think, somewhere in there that stranded that year alive. So we set up a triage team down there, and not just for that year, but every year the turtles start their journey at the Audubon Society down in Wellfleet, where they'll get weighed and measured and logged in. They get assigned what's called an STSSN number. That's just something that allows us to track the turtles. And then they get shipped up to us. So before I leave this slide, I just want to point out that some of the most common questions that we usually get are, why do the turtles go into Cape Cod Bay? Other questions that we get are, of all of the turtles that go in Cape Cod Bay, how many of them make it out? Here's what we know about that. Next slide. There's a lot of unanswered questions. We need to do a lot of research in Cape Cod Bay. There's a lot of competition for funding. It's the Cape and the Gulf of Maine in particular are a rich area for megafauna. There's a lot of whales and whatnot. So there's competition for funding. It's hard to get research projects funded for these smaller sea turtles because they're not a megafauna, but they are important. So there's a lot of work that still needs to be done as to why do these turtles come up all the way up here. Again, the Ridley comes from Mexico, so it makes its way all the way around the Gulf and up and into Cape Cod Bay. So some important things we need to figure out at some point. So I'm going to take you on sort of just a little walkthrough of when the animals come in, what happens to them. I already said that they usually get transported up to us by the Audubon staff and their volunteers. They have a whole group of turtle drivers. And these are folks who are trained. They know when the turtles are in their car and they're coming to Quincy, they can't have the heat on. We're going to talk about that in a minute, but when the turtles strand, you can't just warm them up quickly. It's got to be a slow warming, and Dr. Innis is going to cover a little of that. So you see these banana boxes all lined up here. The turtles are actually, there's one turtle per box. And I don't know, the Audubon staff must have some sort of relationship with a banana or fruit company down there. But every year it's a banana box. It's never an apple box or whatever. So, but yeah, we're trying to get the banana people to donate, but so far nothing. So the turtles all come up. You'll see also on top of those boxes, you can see some folders here. Again, each turtle comes up with its number. So right off the beach, it gets assigned a number, and that number will stay with the turtle until the day it's released. Sometimes that takes a few months, sometimes it's a year. But that allows us to maintain very accurate records of who's who, where did it strand, also its weight, its length, and then that way, if that turtle after release is ever found nesting on a beach in 10 years or at any point, we can track it back through that number. So here's the turtles on arrival. They all come in their boxes. They all get matched up with their paperwork. And then one by one, they come into the exam room. This is what they look like upon arrival usually, or some version of this. You can see it's covered in sand and algae, lethargic. There's a lot of pink down here. Again, these animals are really cold. Sometimes they're coming in 40, 45 degrees. And this is what the lower shell is called a plastron. This is what this looks like, and you'll notice a lot of bruising here, and again, just covered with this sort of algae and other debris. So they'll start with a physical exam. One of the things that's happening here in this picture is because these turtles have hard shells on the top and the bottom, a stethoscope really is just non-functional for them. So we use a fetal heart monitor. It's a little Doppler. And it works very well, and it's one of the only tools other than something more sophisticated like an ultrasound. It's fairly reasonably priced, so we have several of these, and it allows us to figure out what the heart rate is on these turtles when they come in. And they're quite low. You'll hear Dr. Innis talk about that in a few minutes. Now, a lot of folks just, what happens every year is people find these turtles on the beach, and they think they're being helpful, they put them back in the water. That's not a good idea, because again, as the water temperature is going down and down and down, and the turtles don't exit Cape Cod Bay, their resistance is becoming more and more susceptible to anything that they might come across. Maybe it's a virus, maybe it's bacteria. They're getting colder and colder, so they start to slow down on their food intake. They get battered around by the wind and the waves a lot. So when they come to us, these are little kiddie pools. You can see a guy back here, and there's a bunch of turtles in here. This one is actually being supported because it's not strong enough to swim yet. It often takes a few minutes for these animals to animate. So we record when each animal goes into the kiddie pool, and there's always somebody there, a little lifeguard we call them, and they record the activity level of the turtle and how it looks. Is it moving all of its flippers? Does it appear lethargic? Sometimes they'll sink right to the bottom. So these shallow pools work very well for us to just sort of ramp those turtles back up. You're sort of almost retraining its body. But even the weak ones like this, we have to get them in water. You have to get them moving because their heart rates are so low. Charlie will talk a little bit more about that. But if we don't get them moving, then they'll eventually just slip away from us. And usually that will happen overnight if we aren't careful. So I talked earlier about there being 144 turtles one year. Last year, we had 126. So one thing I always worry about is whenever I'm lecturing or Charlie's lecturing, it's never about one person. This is a team effort across the aquarium and across with all of our volunteers. And you'll see other folks even outside in the community reach out to us and help us get this done. So here's a picture of just some work going on in our clinic. And it takes two people for every turtle. One person is holding and the other person is actually performing whatever the clinical procedure is. It might just be collecting a heart rate, or maybe it's giving fluids. But there's always 2 people per person, per turtle, sorry. And just some little action shot there of what's going on. This I put up because this was my Christmas team last year. You know, again, it's a holiday. You always think about spending the day with your family, and I love it that all of these people gave up their Christmas to come in and help us take care of turtles. So that's an extra special group for me. And then we can't talk about rescue without talking about the animal health department. This is Katie Kodzis. We call her Superstar at Running Bloods. When we have a lot of turtles that come in quickly, we take a blood sample every day just so we can run electrolytes. And sometimes there's 20 samples that need to be taken, and Katie is really organized and very fast at doing that. They all are, but she's one that just in particular is quite skilled at it. All right, we're stuck here. Let's try this. There we go. All right. So again, just more slides. I'll put them up all here for you. Of some of the other things that need to get done just to take care of these animals. It's not just about the treatments or about the blood work. All the animals, every animal gets a record, and everything that animal does, everything it eats, everything we do to it, if it's giving it fluids or taking a blood sample, we even record how much blood we took every time we take it. So in the upper left-hand corner there is one of our vets, Julie, who's sitting in the back. I don't know why you're so happy about writing records, but I'm glad. That's always good. And then we also have a lot of volunteers. Again, we had a lot of animals. So this is one of the biologists from New York. We called in some help. We had help from Mystic and several other agencies sent us up. Virginia sent a turtle biologist up to help us. This is a fun picture. Sometimes preparing the foods for their, just the daily diets can take 3 or 4 people 3 or 4 hours of cutting up squid heads and little pieces of fish and that sort of thing. So, and then this is Nina, our water quality expert. Again, we collect samples from the water, and it's tested every day just to make sure it's as clean as we can possibly keep it. So the next thing I'll talk about before I turn things over to Charlie is just the feeding. We often get people who come down who just think you can take a whole squid and huck it in the pool and that the animals are going to eat it. And these are animals that in the wild, they have to hunt for their prey. It's a fight or flight. They have to chase it down. And they are not accustomed to really eating dead food. So we have to train them to eat. We also do it very carefully in accordance with what they can handle clinically. So this is what some of the food looks like. And in the back, this is how they start. So when they first come in, they get what we call fillets. So there's no hard parts, there are no bones, and they're cut fairly small. And we have to sort of get the turtles to start eating those first, and it gets the GI system back up and running. Because again, they've been floating around out in cold water. They haven't been eating. In most cases, they haven't been eating. Sometimes they have. But the GI system isn't fully functional. So they graduate from soft parts into pieces of fish that actually do have bones in them. And usually that's after we've done a second radiograph or an x-ray that shows that there's nothing impacted and we know the GI system is working. Then we graduate them to hard parts. And these are squid heads, which are a favorite of the sea turtles. And generally what we do to get them eating, because if you just throw it in, they usually will not eat. So we use tongs, very long tongs, and you jiggle the food. You got to sort of make it look like it's swimming away from them. And that tends to work. And we recommended this. I went down for a cold stun event in Florida, and they were having a lot of trouble getting these turtles to eat. And I said, well, we need some tongs. So we sent someone to the store, and I said, just buy a bunch of tongs. And they came back with these giant salad tongs that you see at a picnic. And I'm like, wow, no one's going to eat. That's scary. So, and they didn't. But yeah, so these are just small. They're used in they're like surgical tongs or whatever. But they're non-invasive, and if you jiggle the food, they will come and take it. And then we gradually get the tongs out of there. We desensitize them to that, and we do broadcast feed. So this is where the food gets thrown in. You can see a piece back here, and this turtle is actually taking it probably off the ground. And eventually they'll take it right out of the column. So it's not associated with us hand feeding or tong feeding at all. They're now eating again back in the water column and not looking around, looking up for food. All right. So I'm going to turn things over to Dr. Innis, who's going to talk about the clinical part of how we take care of the turtles.
From a medical perspective, we find that turtles that come in when they're very cold are physically debilitated. Their heart rates are very low. Sometimes they're not breathing very well. The immune system of reptiles is very much dependent on their body temperature, and so when they get very cold, their immune system doesn't function very well. And we find that a lot of our turtles are prone to severe infections of the lungs. They get pneumonia, and these can be really difficult to clear. So a lot of what I want to show you today is some of the techniques we use to get the turtle stronger, and some of the tests that we use to determine exactly what's wrong with the turtles. Over time, we've found here, and other studies too have shown that you can mess up the chemistry of turtles by changing their temperature very quickly. And so even though these turtles come in cold and you might think that you should just warm them up to get them back to normal, if you warm them too fast, their bodies can't keep up with that. And basically all of the functions going on in your body are just chemical reactions. And if those chemical reactions are deranged enough, that can kill you. And so if you derange those chemical reactions even more by trying to challenge the turtle's body by getting warmer and warmer and warmer very fast, you can cause further problems. So we actually find that it works best to only warm them up by about 5 degrees per day. And so if they come in at 50 degrees body temperature, they may not be up to 75 or 80 degree body temperature where we want them for 5 to 7 days. But that's okay. They actually do better that way than if we just instantly put them under a heat lamp at 80 degrees. When they come in, we do a physical exam, as Connie mentioned, which tells us about their heart rate and respiratory rate and other things that may be injured. I'll show you some images of some of the injuries that we see. We also collect blood from them, and there's a whole series of blood things. I'm going to show you a tiny bit of data, not to overwhelm you, but just to kind of prove the point that there's a lot you can learn from looking at turtle blood values. And we give them fluid therapy. A lot of times they're dehydrated, so just like a marathon runner that's injured, that's dehydrated after a marathon and gets an IV, we give these turtles sometimes IV or more routinely subcutaneous injections. I think we'll do questions all at the end, okay? And because of the infections they get, we sometimes need to use pretty heavy-duty antibiotics or antifungal drugs even sometimes. So when they first come to us, we see injuries from the stranding. Sometimes they've also been injured by things like seagulls on the beach that start pecking the skin off their head or ripping their eyeballs out, things like that. One thing that is always a little bit fun when they first come in is determining what species of turtle it is. And usually it's pretty obvious what species it is, but every once in a while we get in a turtle that isn't quite right for any of the known sea turtle species. And there actually are hybrid sea turtles out there that are mixes of other sea turtle species. We had one this year that we still don't quite know what he was, but he didn't look like any of the known species of sea turtles. Temperature, pulse, and respiration is obtained when they first come in. We weigh them so that we can calculate their drug doses very precisely based on their body weight. Looking in their mouth is important. There's a lot of stuff in the mouth. The opening down into the airway, the opening to the esophagus, their tongue. And this is an area where we can see some types of infectious diseases in turtles. This word coelomic is sort of like abdominal. Reptiles don't have a diaphragm, so they don't have a thorax and an abdomen. It's all one big body cavity that's called the coelom. And so we use this term coelomic when we're feeling the turtle's belly, sort of. It's technically the coelom. We also feel their legs. We do see some turtles, and I'll show you an example later, that have orthopedic injuries of their legs. So this is a turtle getting the heart rate taken, as Connie said. And usually if the turtles are pretty stable, we can detect their heart rate with this Doppler monitor. This just creates a swooshing sound every time the heart beats. And turtle heart rates can be very low normally, and they're even lower when they're cold. So it's not unusual for us to have a turtle whose heart beats only once per minute when they first come in. And so you have to sit there with the probe on them, waiting and waiting and waiting and waiting, and you're wondering whether they're even alive. And then you might hear this gentle swoosh that tells you that at least their heart is still functional. if you can't hear the swoosh, it doesn't mean they're dead either, because it could just be that their pulse is not strong enough to create the swoosh. So if we're still not sure if they're alive, we go to ultrasound, and what we see on the top up there is basically an echocardiogram, and the red is indicating that there's blood flowing through the heart when the heart contracts. So we can have a turtle come in where there's absolutely no sign that the turtle's alive. They don't blink when you touch their eyes. They don't care if you stick your finger in their cloaca. They don't care if you pinch their toes really hard. And they're just there completely non-responsive. And you put the Doppler on and you don't detect a heart rate, but you put the ultrasound on and you wait there for a while, and all of a sudden you see this little flutter of their heart muscle. And some of those turtles will survive. And so we keep treating them until we either can't find any heart activity at all, or if they just become stiff and they're clearly dead and they're in rigor mortis. This is one of the eye injuries we see. This guy got poked probably by a seagull right through his cornea there. So we always look for that initially. Blood sampling, we usually get blood samples from the jugular veins, which is pretty easy once you learn to do it. A lot of these turtles are not breathing when they first come in, and because of that, if you don't breathe, your oxygen goes down, your carbon dioxide goes up, and that creates all kinds of problems, and we can detect that on our blood tests. And so we treat those guys by connecting them. We put a tube down their trachea, just like if you're under anesthesia or on a ventilator as a person. We connect them up to a ventilator. We actually had to customize our ventilator because most ventilators don't go to the really low respiratory rates that you want for sea turtles. If you put a turtle on a human ventilator and breathe for them 10 times per minute, that's actually hyperventilation for them, because they may only breathe once every 5 minutes or 10 minutes or 20 minutes when they're diving. So it's easy to screw them up if you breathe too much too. This initial swimming, Connie mentioned it gets their blood flowing. That's true, and it's important to she always says it reminds them that they're a turtle. We think that if you have a turtle that's debilitated and you just let them sort of remain inactive, they really don't have any stimulus to start becoming turtle-like again. But sometimes if you put them in water, they start to feel a little more turtle-like and they start moving, and that helps their blood to flow. It also helps to get all this crud off their shell and their skin. And if they drink any of it, this is just pure fresh water at this stage. It helps to hydrate them. This is just a quick example. I'm not going to show you too much on this, but we've done a lot of data analysis over the years of hundreds of turtles that we've brought in, and we're actually getting pretty good at being able to look at the blood on the day that they come in and almost be able to know whether that turtle's going to live or not, just from the first blood sample on the first day that they get to us. So just as an example, if you look at the top number there, sodium value of 180, normal is in the 150s. Of the data we looked at, this was 5 years worth of turtles. We had 7 turtles whose sodium was 180 or greater when they came in, and none of them lived. We had 27 turtles whose sodium was greater than 170 when they came in, and 17 of them died. So that's about half of them. So we're getting pretty good about saying, your sodium is greater than 180. It's very unlikely that you're going to live. It's not to say that we're going to give up on you, but if we have 20 turtles come in all at once, all on the same day, and 19 of them have pretty good sodium values, and one guy has a sodium of 180, we're not going to focus all our attention on the 180 guy, because it's very likely that he's going to die anyway. It gives us a way of deciding where we're going to focus our effort, so we can put our effort on the guys that are most likely to survive and not on the guys that we know are pretty likely to die. Fluid therapy, this is putting a little needle through the skin, and we inject fluid under the skin and they absorb that. That works really well. This is some work that Dr. Hunt here has done. Some of you came to her whale poop lecture a few weeks ago, and this is some turtle stuff that we're doing. It's not a surprise. You might imagine that a sea turtle that's stranded and washes up on a beach might be stressed. And we always think of stress as just, oh, I'm stressed, I have so much going on. But there's actually physiological stress that's bad for you. And Dr. Hunt has shown, when we've gone back and looked at the blood of our turtles, if you just take these as a group, turtles that when they first come in, these are some of the highest stress hormone levels that have ever been recorded in sea turtles for our group. All right, and we know that that suppresses their immune response, among other things. for the guys that do well and end up getting better, this is their stress hormone level when they're nice and happy and they've been in rehab for a while and they're getting better. So it's kind of intuitive, but it's nice that we've actually been able to prove this now. We take a lot of x-rays of turtles. This is kind of just showing what a normal turtle x-ray looks like, and I want you to focus on this area here that's kind of a uniform gray, and out here it's kind of a uniform gray. That's their lung field. So get that image. All right, and then this, you see this got kind of these honeycomb kind of look throughout? All right, so this is what we see in a lot of these guys that get pneumonia. And in the guys that die, you can actually see in here, these are turtle lungs. These are fluffy white fungal colonies inside the turtle's lungs. So this is a fungus that's causing pneumonia in these guys. And so you can imagine if you have stuff like that in your lungs, you're going to be pretty sick and it's going to be hard to treat. So to determine these, this is a turtle having a CT scan. This is out at Mass Vet Referral Hospital up in Woburn. They let us do CTs. And it's nice because we can get these different views, and it gives a lot better detail than when we get on an x-ray. And one of the things we've been doing for the last few years with these CTs is we can very precisely measure. So like in this view, the turtle's tail is over here, his head is over here, and you're looking at the side of him. And this is his lung field right here. So if we know we want to go after this area of pneumonia to find out exactly what type of infection is in there, we can drill a little hole right through here through his shell and go in and sample that. Or if we know that we want to go down here, we can use a scope and go in front of his leg and sample the lung right there. So these little scopes that we use, these are really cool. They're only a couple millimeters wide. You can see how small they are. We can make little holes in different parts of the turtle's body, put the scopes in. And we have these little tiny instruments that you can put down through the scope and take samples. So if we go in the turtle's mouth with a scope, we can go down his esophagus, or if we go down his tube here, this is going into his trachea that would lead us down to his lungs. This is what a turtle esophagus looks like. They have these little, well, sometimes big. These are made out of keratin, like your fingernails. And it's designed so that the turtle can ingest a bunch of food and a whole bunch of salt water, and then they can expel the water, but all the food gets caught on these things, and then they can swallow all that food. This is what we see when we do these scope things down the trachea. So this is the turtle's trachea here going down. Eventually you get to where the two bronchi split to the lungs. And if we go further down, we end up in the lung of the turtle, and then we can use our instruments to sample down in there. If we can't get it that way, we can make an incision here. This turtle is right here, his head is over here, his tail is right here, and his hind leg is down here. And we're making an incision basically like right in front of his hind leg. And then we put our instruments in through here, and we can see inside the turtle. All right, so this is inside a turtle having a scope procedure done. The procedure itself isn't important, but I just want you to see how this goes. So we've got a scope in, and then we've got another veterinarian next to the scope who's grasping the tissue we need. And then these scissors are cauterizing the tissue. And this is how we can get samples from the inside of a turtle. When they're totally surrounded by the shell, we don't need to cut any of their shell. We can just go in through these little skin incisions. And then the scissors are going to cut this tissue free. And if this were something like an infected area of lung or an infected piece of liver, we can just snip it out. And remember the size of these instruments. They look gigantic here, but these are those little guys next to the penny that are just about 2 or 3 millimeters in diameter. Okay, another way we can do this is drilling a little hole through the shell and then putting our scope down through that hole. And when we get in there, these are some of the areas, like these are little areas of infection in this turtle's lung. Okay, one of the things we treat turtles with that have pneumonia, in addition to injections of drugs, is nebulizing. So we put them in a chamber and we nebulize antibiotics so they're breathing in the antibiotics. I'm going to skip this. This is a little turtle that had acupuncture, actually. He had a disorder of his jaw, and we ended up having acupuncturists come in and treating it almost like a person with a problem with their TMJ joint, and it worked great. He was released. This is a turtle, a trauma case, just a before and after. We get these guys sometimes that are hit by boats and their shell is cracked. This guy also had a broken leg, and it cut through the artery that supplies the end of the flipper. So he ended up with kind of losing the hand part of his flipper, but he had the rest of his flipper. You can see his shell healed up really nice. He was released. This is a case that had infection of his shell, and the shell all died off. And eventually we were seeing this, and when he breathed, this was just the surface of his lung that was moving up and down. And so we ended up using this experimental compound made by Pfizer. They were guys here in Boston brought it over, and we injected this area with a thing called bone morphogenic protein that stimulates bone formation in soft tissue. This is him a few weeks later. It's filling in with normal shell. It's a few weeks later, and this is pretty much close to the end when he was released, but this was all that area that was exposed lung tissue, and now it's healed in with bone. Okay, in general, these cold stun turtles need about 7 to 9 months of rehab. Our success rate, depending on how we do the statistics, is somewhere between 70 and 90%. And just as an example, 2010, where we had 113 Kemp's Ridleys, we know that we released 88 of them back out into the wild. All right, we're going to zip through some leatherback stuff here next.
Yep. I'll make a quick swap again. Okay, so I'm going to talk a little bit about the leatherbacks. You saw some pictures earlier, at least that one in the ID slide. And the leatherbacks are critically endangered. The Pacific leatherback population, there's some estimates going around, population models, that are putting that population in such peril that it might be extinct within the next decade. That's kind of a scary thing. The Atlantic leatherback population is doing a little bit better, so that's good news for our side, but it's not good news overall. So the leatherbacks essentially were swimming around when there were dinosaurs around, and that's really true. They've been on this little planet for over 100 million years, which is amazing. And when you look at some of these pictures, you'll see some close-up of their faces. They look prehistoric. They really do. You'll never see anything like them. They are the largest living turtle in the world. They can weigh in over 1,000 pounds. And Dr. Innis and I have worked with a few, you can see down here in this picture, with some that were just monstrous. And there's no real controlling them when we're working on deck. They just kind of move around a little bit, and we move with them. There's no telling them no. So the leatherback grows pretty much faster than any other reptile. It's also the fastest swimming sea turtle. The photo that you saw just a minute ago, including the animal itself and the flipper span, that can be 10, 12 feet. So we end up getting bruised and banged up a little bit on the boat when we're working with these animals because they're so enormous. And again, they've got those giant flippers that are kind of flapping around. One actually broke my nose, so it wasn't pleasant. But as you can imagine, these are large animals, giant flippers. They're very deep diving. There's records of them diving over 4,000 feet. That's just amazing. That's equal to some of the great whales that are out there. Well, you'll see towards the end that these animals are capable of long pelagic migrations, just crossing the oceans. It's amazing how far they go and how deep they dive. They're also subarctic. As we've been talking tonight about cold stunning and how we have to raise the temperature of these little hardshell turtles slowly and carefully, that's not the case with the leatherback. The leatherback is capable of generating some metabolic heat, so they don't have the same situation that the little hardshell turtles do. They're subarctic, and there was actually even a record I found of one that was stranded in Alaska. And just to think about a turtle, a reptile in Alaska, it just doesn't seem to make too much sense. So in order for them to dive as deep as they do and to move around, is this here? The way they do, they have some special adaptations in their bodies. Again, look at the size of this guy, he's just a monster. But they have very vascular cartilage at the end of their bones. And that may help in some of the, with the pressure as they're deep diving. You can see on the top of their shell in the upper picture, it looks very smooth, and it is. It's a leathery sort of skin that covers the shell. You can actually push it and put your nail in it, and it will abrade quite easily, unlike the other turtles you've been seeing in the lecture that have the bony shell. So, let's see, sorry, lost my train of thought. the photo that you see down here, an interesting thing about the leatherback is when they die and once they start to decay, I know it's gross, but their shell has these little mosaics, again, probably to facilitate the deep diving because it allows some movement. So as this animal decays, this whole shell will fall apart into little pieces. It's almost like a puzzle. You can actually collect it, put it in a bucket, and walk away with the entire animal that started at 1,000 pounds. So again, they have these special adaptations. They're very flexible, probably to account for that deep diving. Now, these are threats to leatherbacks, but also to all sea turtles. And you'll notice that any sort of human activity is first. And that's sad, but that is the case. Boat strikes, obviously, this year, there's just no other part to the animal. So that was severed probably by some sort of very large ship. But in addition, the other sad thing is that wasn't the only encounter with humans that this turtle had. These marks here are very common. We find those whenever an animal has encountered vertical line and has been struggling to get out, it abrades that skin and leaves that type of scarring. So most likely that animal encountered 2 different forms of human interaction. These photos in the bottom, a little bit upsetting to see, but this animal's obviously entangled, severely entangled. And depending on where these animals get caught in the vertical line, as the tide comes back in, oftentimes they get caught in shallow water because that's where the pots are being placed by the folks who are fishing. As the tide comes back in and the water rises, if they can't get to the surface to breathe, they end up usually in our necropsy room. So it's a bad situation. They start to struggle and they often get quite entangled. The line just keeps getting wrapped around as they spin and try to break free. They end up actually entangling themselves worse. And then the other thing is the boat strike. It's very common. We see it in leatherbacks quite frequently. And it's a good thing this picture is dark because it's very upsetting. This animal actually stranded alive. There's a strike right here through the face, another one through the shell, and another one here that ripped it open. Which leads me to believe that it must have been surfacing when it got hit just because of the direction of the prop strikes. So that animal came in and was euthanized immediately because there was the prop strike that hit the face wiped out both of the eyes. So there's really nothing that we could do there. There is obviously natural disasters. I guess the storms in the south or in the nesting beaches, I should say, not just the south, but anywhere the animals are nesting. Hurricanes can be devastating on the eggs. So there are a lot of factors that the sea turtles need to overcome. Natural disasters being one of them, and then also disease. Charlie just showed you some pictures of pneumonia and whatnot. So one of the things we do is not just work with the animals that strand and come into us, but sometimes we go out looking for trouble. And this project here that you see in the photograph is a project we're doing with the University of New Hampshire, UMass, and the state of Massachusetts. And we actually go out and we look for leatherback turtles. We have a spotter plane that flies above us and he finds the turtles. It's fairly easy. It's easier than you think. If you can find the jellies, you'll find the leatherbacks. That's the exclusive food source of these giant turtles, which is amazing because there's not much nutrition in jellyfish. So once we get out there, we have a procedure to collect these animals. Essentially, we are catching them in a giant butterfly net and bringing them on board. And you'd never think it would work, but here's some proof. And what's happening is some of our colleagues, actually one of our colleagues is going to be presenting here in a few weeks on her data from the satellite tags that she puts on these animals. But Dr. Innis and I have been taking a lot of samples from these animals, biological samples and whatnot, so that we can try to figure out what normal values might look like for a leatherback turtle. And we've been quite successful in that. In our history here, we've had 2 live leatherbacks that actually stranded with severe injuries. And it was very difficult for us to take care of it because there wasn't a lot of published data on what their levels should be. You know, as Charlie mentioned earlier, gave you an example of the salt level in turtle if it's over 180. Well, we didn't know what that meant in a leatherback. But now we do, thanks to this study. We've collected a lot of information. Do you want to talk about some of these?
Sure.
Going back and forth on you a little bit.
I'll be fast so there's time for questions. I know it's close to the end here. When we bring the turtles on board, either if they've been entangled or if we've captured them directly for part of the study, we try to keep them as calm as we can, although they usually are very resistant to that. But it does help to put these damp cloths over their face, and it seems like just reducing the visual stimulus is helpful. So we measure them. We can't really weigh them because they're so large, but we do get measurements. We take this little flexible probe and put it up inside their tail to get their core body temperature. And to get their heart rate, we can't really use that Doppler very well up on the neck like we do in little turtles. Their heart is just too far away from that. So we found using an EKG sometimes gives us a little readout like in people, or we can put the Doppler probe sometimes aiming up under their shell towards their kidney. For blood, we get it out of the jugular vein. It's just much deeper, and so we often use a needle that's about 3 inches long to get all the way to the jugular vein. Or we can get it out of their tail. This is my left hand holding the tail of a big male leatherback, and we can get blood from the top of the tail sometimes. As Connie mentioned, one of the cool things about leatherbacks is that they can keep their body temperature higher than their environment, and that's really not true of any other living reptiles. You can see here the body temperature, the average of the turtles we caught in the Cape Cod area is 26 degrees Celsius, or about 75 degrees. Where the sea surface temperature is quite a bit cooler than that. So they're actually generating heat by their muscle activity. And this is just showing we've been able to compare the blood of entangled turtles to blood of healthy turtles that we're capturing and demonstrate some of the derangements that you see in entanglement and maybe what's killing them when they get entangled. One of the cool ones that surprised us was this one. This is called Beta-Hydroxybutyrate. It's something that our bodies produce too when we're fasting or in a negative energy balance. It helps to produce energy. You can see healthy turtles have very low BHB levels when they're out and feeding normally. And this is the average of turtles that are entangled. When you start to get numbers like that, at least in dairy cattle, it means that you're going into this downward spiral of metabolic energy metabolism, and that can kill cattle, and we presume that maybe going into something like that is one of the ways that leatherbacks die when they get entangled. Their stress hormones on the right are much higher in entangled turtles than turtles that are not entangled. So you would think that going out and catching a turtle in a big butterfly net is stressful, but it's really not that stressful. But getting wrapped up in fishing ropes is a lot more stressful, and that creates further problems for them too. So this is our most recent live leatherback. It was brought in from a beach in Dennis, Mass. In November. And we brought it into the clinic. Unfortunately, only survived about 24 hours, but we had it on the ventilator and all this good stuff. This guy had rope entanglement injuries. The ropes were gone, but this is his front flipper. This is another front flipper, and this is all the way down. The rope has cut through his skin all the way down into the muscle. One of the things we really try to do when we get a lot of information together finally is publish what we get so that we're not the only ones in this little building here that know what we have. We can publish it and hopefully stimulate other people to publish their findings. So this is just a handful of the stuff that's come out of our 2 departments in the past few years. Connie's going to talk about what happens when we let the turtles go, and then we'll be done.
Oh, okay. All right, thanks. All right, so we're nearing the end, and we'll try to let you out on time. After the rehab is finished, the best part for us, obviously, is the release. It takes quite a bit of coordination. We have to work with the town because there's security problems. People will mob the beach, so we kind of have to work with the town, then we sneak in there, and whoever happens to be on the beach that day gets a heck of a show. So you can see all the feet in the background. This was just a couple of years ago. Some of these animals have tags. I'm going to talk about that in just a minute. But, you know, again, there's a lot of long hours when they first come in. Sometimes these are 10, 12, 14-hour days, and you get up and you do it all over again. So this is really the best part, and we try to invite everybody that helps us take care of these turtles, from the drivers that bring them back and forth to folks that help maybe fly them south, and I'll show you that in just a minute. So they're quite, they're a lot of fun. And here's a green turtle that's kind of crawling down the beach. This is a lot of debris on the beach, shells and things, but he's carrying a satellite tag on his back. And I'm going to show you some maps and talk a little bit about the type of data that we get and what we learned from this. It's really important. We're learning things that we didn't expect to learn. Sometimes we're released from boats. Last year was the first time we did that, and it was a lot easier. It didn't take quite as much coordination, but we lost the crowds. You know, we were on a boat, there was only a few of us, and we realized, like, oh, yeah, you know, congratulations. But this is such a learning opportunity. And one of the things that we have to remember and that we have to do is get other people interested in turtles. If none of you showed up tonight, Charlie and I would probably still be doing this because we're turtle geeks. But we need everyone to be turtle geeks. So I think we'll probably go back to doing this just because it's a lot of fun for these folks, you know, to join us, and they're learning. And we're also trying to encourage an appreciation for sea turtles. So many folks tell us that they didn't even know turtles were in our area. So we're getting a lot of star power now interested in sea turtles. This is best-selling author Patricia Cornwell. She writes the K. Scarpetta series. She came out with us last year on our turtle release and really enjoyed it. And then we had Spider-Man show up. That was really smaller than I expected, but I guess he works it off fighting crime. There are turtles that are ready to be released before our water temperature will permit us to release them. So if our water temperature here is still too cold, for example, right now, we have 10 or 12 turtles that are ready for release. Oftentimes what we'll do is we'll ship them south by one means or another. We work with a lot of partners. We're part of a large network. Here's the truck packed with turtle boxes. They're all secured in their little boxes. And this is us with the National Aquarium in Baltimore sea turtle team. It was warm enough around them at this time whenever we did this release, and so we went that far and we released with them. A couple of years ago we had a lot of turtles, and we had a crew that was ready earlier, and the warmest water we could find that, you know, was appropriate for the species we were trying to release was in Georgia. So we contacted those folks. We went down to the Georgia Sea Turtle Hospital. The nice thing about that is they carry all your stuff, so you can just kind of hang out and take pictures. So we pretty much do whatever it takes. We often have to fly them, depending on our resources. If we still have a lot of turtles back at the ranch and we can't really, we need all our staff there taking care of the animals, we can't afford to put 2 people in a truck to drive south, we'll reach out. This was a sweet ride, but it's very rare. And this was last year when we had 126 animals. We reached out to NOAA, who reached out to their partners at U.S. Coast Guard. And these folks were great. This was a really bad flying day, middle of a storm, but they flew. This is the crew here. These guys are just phenomenal. They helped us load all the turtles. They were just great. More commonly, we use folks that are in the Civil Air Patrol, and that works out really well. They have some funding that they can help out, so they oftentimes will absorb the fuel cost, and they're all volunteer pilots. So it doesn't cost us anything when we fly them with the Civil Air Patrol. So I can't say enough good about them. They made several trips for us last year when we had all those turtles. This was a new relationship we had this year. It's somebody who's on the board of the South Carolina Aquarium set us up with this company. They happen to be up here in the area doing demos with this new plane. It's a new aircraft, uses much less fuel, it's lightweight. And they said, sure, we'd be happy to fly turtles for you. So we made sure they got a lot of press. There was 1.5 million people that saw this story just in the first day. So that helped. Maybe we'll use them again. More commonly, the 2 guys are 2 go-to pilots. Owen Carr, he owns this plane. He's done a number of turtle flights for us. It's a small plane, as you can see, so it's a little limiting depending on if there's a cold front moving through. I now know more about airplanes and takeoff and runway lengths than I ever thought I would know. But all of that is important, and it's safety first. We've aborted several flights the morning of because the weather wasn't good enough. This is one of our other pilots. This is Mike, and he's probably done more flights for us than anyone else. We haven't hit him up yet this year, but we probably will. But again, that's his own aircraft there, and he donates all of the fuel. And the cost of Jet A these days is pretty outrageous. So I wanted to just quickly show you a little bit of the data that we get from the satellite tags. Here's the tag here. This is a green turtle. It's a tiny little tag. This tag weighs probably about 200 grams, anywhere in there, sometimes a little bit more. And we always keep a pretty small platform because we don't want to increase drag on these turtles. And so this is a map. There's multiple turtles on this map. Each one is designated by a different color. This is a green sea turtle, not this particular green sea turtle. But this is one green sea turtle that went up here. This is Canada. And that is absolutely outrageous. It's something we never would have predicted, and we never would have known if we hadn't applied a satellite tag to this animal. We were just less than a month of 2 years, a month shy of 2 years of data on this animal, which is a really long life for a sea turtle tag. So we learned a lot of information about that, and this is a typical behavior. When he tucked in here, that was a hurricane. And we've seen that multiple times with turtles that have been carrying tags that we released, that within a few weeks later, a few months later, a hurricane came along, they tucked right in like that. So we're just going to look, I just picked two so you could see it's a little clearer than, you know, having multiple on there. Here's a green turtle that worked its way right down the coast. The tag stopped right down when it got off of Florida, but he just hugged the coastline. And yet here is that guy we just talked about, way out here in the Gulf Stream where it's nice warm water. So we don't know why they're doing this. We have had several turtles come out here and overwinter in the Gulf Stream, which again is something we did not know. So we actually just got a sizable grant to satellite tag more turtles. We got $40,000. And of course this year we had no green sea turtle strands, so that's just how it works. But we're really curious about the green sea turtles since they're the ones that seem to overwinter out here, which again is just something we didn't know before. And this is it's a little teaser slide, we like to call it. These are tracks that are going to be presented by our colleague Cara Dodge in a couple of weeks. She's coming here to present at the Lowell lecture series. And she's our colleague that works with us on the Leatherback project. And she's going to be talking about some of the data she gets from the dive depths that she has from her tags, and as you can see, how far they go. I mean, this is just unbelievable distances that these animals are doing, but I don't want to steal any of her thunder. So with that, we will open it up to questions, and we'd like to thank you for all coming. Thank you all for coming tonight.